Good afternoon. Welcome to week number 10. How many of you are excited that it's week 10? How many of you are disappointed and wish we had like 16 weeks in this course? That's what I thought. Okay, I'm just checking. So what we will do today is finish our discussion of enols. We'll talk about enamines with any residual time we have. We'll start our last topic, which is pharmaceuticals. Later on in this week, we will begin to talk about the final exam. We'll do that on Wednesday. So let's review where we were on page 64. We were talking about enols. Enols are isomers of carbonyl compounds in which we have switched the position of the pi bond and a hydrogen. And so the structure looks like this. There's our carbon-carbon double bond and an OH. And the isomer, the tautomer, which has the carbonyl, is called the keto form, not because it is part of a ketone, but simply because it has a carbonyl in it. Any carbonyl containing one of these is the keto form. So it can be a ketone, aldehyde, ester, carboxylic acid, etc. They can all also have an enol tautomer, and that's the one where we have conceptually shifted the alpha hydrogen over to the oxygen and the pi bond over to a carbon-carbon pi bond. This tautomer is called the enol form because it is an alkene and an alcohol, enol. The interconversion of these two is called tautomerization, and in general, for simple cases where there's no special stabilization for the enol, the keto form is more stable. So, for example, in the case of acetone and its enol, the keto form, the equilibrium favors the keto by about 10 to the 6. And that is because, in simple cases, it's simply the bond energy issue. If you were to add up all the bonds that are present in the keto form, add up all their bond strengths, compare that with all the bond strengths that are in the enol form, you discover that the keto form is a few kilocalories more stable. However, if you add elements to the structure, I mean pieces, not elements as in plutonium, but pieces to the structure that make the enol more stable, now the equilibrium can shift. It's not because you're making the keto form less stable, but you're improving the stability of the enol form. So example, in this 1,3-dione that we have here, pentane dione, the enol form, there's the enol form, has two things which help stabilize it compared to the acetone enol, and that is an intramolecular hydrogen bond as well as conjugation. So the more conjugation than the acetone enol has. So the combination of those two make this enol relative to the keto form more stable than in the acetone case, so the equilibrium constant is closer to 1. Here it's not exactly 1. It depends upon temperature and solvent and a few other things, but it's much closer to 1 than it is for the acetone case. Now we can go to an extreme case for this keto-enol equilibrium. This enol is present by a huge amount, almost 100 point as many zeros as you'd like, because this enol has something this keto form doesn't have, and that's aromaticity, which we know is worth tens of kilocalories of stabilization. Now, most of the time we won't think of phenol as being an enol, but in fact it is an OH attached to carbon-carbon pi bond, so it does have some enol kind of properties to it. And we also talked a little bit about how the tautomerization process works. How does a keto form become an enol, and vice versa, how does an enol form become a keto? There's three ways in which enols show up. One is acid-catalyzed tautomerization. This by far is the most common, most important mechanism for tautomerization. In this, we start by protonating the carbonyl. It requires a strong acid to protonate the carbonyl, because after all, we're protonating a carbonyl. So the protonated carbonyl can be thought of as a carbocation. There's the resonance contributor. That's one way to think of it. Of course, this is not the most important resonance contributor, the oxonium ion structure is, but we can think of this as a carbocation, and if we think of carbocation fates, being deprotonated to form a pi bond is one way to lead to the enol. Or you can simply take the oxonium ion structure and deprotonate that and go to the enol. That deprotonation regenerates your H3O+, so that tells us that the acid here is a catalyst. It's used in the first step and reproduced in the last one. And we can go in the opposite direction as well. Take a carbon-carbon pi bond, put a proton on it to give a carbocation. What do carbocations do? Resonance, deprotonate the oxonium ion, and you're back to the keto form. So that's one possibility. Another possibility is base-catalyzed tautomerization. So instead of using a molecule of water for the proton shuttle, we're going to use hydroxide for the proton shuttle. Take the proton away and make the enolate. Draw the resonance contributor. Now we can put the proton on the carbon, which goes back to where we came from, or we can put the proton on the oxygen, which goes to the enol. This process, while we can envision it happening, it turns out it usually isn't all that important, because the kind of processes in which you would have base present to make the enol to make the enolate, while this is probably happening, the enolate goes on and does enolate chemistry, like adds to more of the carbonyl compound it came from, before the enol can go on and do anything. The enolate is much more nucleophilic. It's got a formal negative charge. The enol doesn't. So you can draw this pathway, but it's usually unimportant. The third possibility that shows up occasionally is decarboxylation. This is a mechanism step in which a carboxylic acid, which has a carbonyl of some sort that is on the carbon next door. Here it's a ketone, but it doesn't necessarily have to be a ketone. It could be an aldehyde or an ester or any carbonyl-containing functional group. And through a process which probably starts with an intramolecular hydrogen bond, there's a 6-membered ring transition state with 6 electrons moving around in a circle, which causes the loss of carbon dioxide and makes the enol. So this shows up in a few circumstances. A couple of biological kind of processes do this. All right, so that's how enols can be made, and again, acid catalyzed, the most important of those. Now that we've made an enol, what do we do with it? If you remember back in the beginning of this whole topic when we started talking about enolates and so forth, I mentioned that the enolates, enols, and enamines are all similar. They are nucleophilic at the beta carbon. We saw that by looking at the resonance hybrids. In all 3 cases, you could go back and see it on page 54, I think is where it might be. But there is in each case a delta minus on the beta carbon. So that suggests that they are nucleophiles. And that is true. All 3, that's why we're lumping these 3 together. They all behave pretty much the same way. So enols, like enolates, are nucleophilic at the beta carbon, so they can react with an electrophile. What kinds of electrophiles do they react with? It turns out because enols are less nucleophilic than enolates, they don't react with nearly as broad a range of electrophiles as enolates do. But there are a few things that we might recognize. For example, we can make the enol of acetone. Here's our acid to do that. We can make an enol of acetone and have it react with a ketone, more of the ketone that it came from, as a matter of fact. And the net effect is a aldol product. Now, this you might say, wait a minute, that's acid. How can this be aldol? Aldol doesn't tell us. Aldol doesn't necessarily say the mechanism. Aldol simply says what the products are. So we can have an acid catalyzed aldol in the same way that we had a base catalyzed aldol. Now, what's the mechanism for that process? Well, there's certainly no enolate involved, although you might think that. You might say, look, we're making a new bond right here at the carbon next door to the carbonyl. That, by the way, is a hint that you have most probably an enolate, enol, or enamine. There are a few other reactions that we've seen that could potentially do reactions there. But for the most part, if it's a reaction at the carbon next door to the carbonyl, it is most probably an enolate, enol, or enamine. This is definitely not enolate because we're using strong acid. Enolates require base. And it's not an enamine because there's no nitrogen. We'll talk about enamines later on today. So it must be an enol. So let's make an enol. Here's our enol. This is like we saw in just the previous page. The mechanism to make the enol is you protonate the carbonyl and then deprotonate on the alpha carbon. That makes the enol. Now the enol is going to be the nucleophile. So it can react with another molecule of the carbonyl it came from. Remember when we talked about enolates, we say how to identify whether or not you're going to do self-condensation. Well, there's no second electrophile written here in the reaction, so self-condensation must be our intention. And enols, not being terribly powerful nucleophiles, need to have the carbonyl protonated first. So there's our nucleophilic attack on the protonated carbonyl, which we made here in the first step. So that protonated carbonyl, some of it gets deprotonated to make the enol, some of it hangs around to react with the enol. So there's our nucleophilic attack on the enol to give a tetrahedral adduct. The carbon that used to be sp2 is now sp3. No leaving groups under these circumstances, unless you want the enol to leave. But there's nothing that can leave. The methyl groups can't leave, etc. So what do? We're going to move the mechanism forward. Now here we're going to deprotonate the carbonyl. And then we can take, because we have acid present, we can protonate the OH, make it into water, and have it leave. Here we have it leaving in an E2 kind of process. Now, you might feel a little uncomfortable with that because, well, what are our requirements for E2? Good base and good leaving group. And neither of those are present. Water's not a great leaving group. It's okay. It's a moderate leaving group. And water is certainly a lousy base. But we can draw this. This is allowed because what does this structure have? What do you think would be going on, would be being gained in this transition state for this last step? That would make this otherwise not so great E2 allowable. What makes it easier? Resonance. Thank you. Close. It's a resonance idea. Resonance causes this other thing to be a stabilizing feature. Conjugation. Exactly the same reason why we could kick out hydroxide in an E2 sense in the base catalyzed aldol process is conjugation. So overall, it's the same product. Differs a little bit in the nature of the nature of the nucleophile and so forth, but we can still have the acid catalyzed version of the aldol. Now, something else we can do is react with other electrophiles at the carbon next door to the carbonyl. Here's an example. We can put a bromine there by using Br2. That's an electrophile that we've seen reacting with alkenes before. There's aqueous HBr present. That's to make the enol. So you might ask yourself, number one, what's the mechanism for this process? Now, I'll give you a hint. The reaction is occurring on the carbon next to the carbonyl. That means probably, not a guarantee, but probably, it's either an enolate, enol, or enamine. One of those three. Not a guarantee, but again, a good guess because it's chemistry happening on the carbon next door to the carbonyl. All right, so what are our choices? Enolate. Is enolate going to happen under those conditions? Well, what about aqueous HBr? That is really HBr, strong acid. You throw that in water, you get H3O+. Is there strong base going to be present to make enolate when you have H3O+ running around? No. Strong acid and strong base don't exist next to each other. So that rules out enolate. That generally requires strong base. What about enamine? Now, if you don't remember, here's the structure of an enamine down here in the bottom of the page. It has something in this structure which is completely absent in this reaction that tells us it can't be an enamine reaction. Nitrogen. So that means it's got to be an enol, and that's what the aqueous HBr does. It makes an enol. So this is an enol mechanism, so that's one thing to work out for yourself. Draw all the curved arrows. And then a question to also ask yourself is, why does the bromination occur at the CH2 on the left-hand side here, so to speak, between the benzene ring and the carbonyl? Why does it occur there as opposed to the methyl group? I'll leave that for you to figure out. Those are good things. By the way, this issue, if you're looking for the answer, the mechanism is in the OCATSA chapter for enolates, enamines, and enols. All right, let us shift now to talking about enamines. Enamines are alkenes that instead of having an OH on, they have an amine on them. And they are usually, here's a typical enamine structure. There's the alkene with the amine attached. Most of the time, they're formed from a secondary amine. That's an amine that has two R groups and one hydrogen, reacting with an aldehyde or a ketone. I can't make them from esters. Why can't I make an enamine from an ester? What would happen if I took an ester and reacted it with a secondary amine? What do you think the product of that would be? Why? What I'm saying is, if I take, let's say, this guy, and I react it with a secondary amine, dimethylamine, according to the scheme that's up there, I might expect to get this. So where was the carbonyl becomes the carbon-carbon double bond and the new carbon-nitrogen bond. But in fact, that's not what happens. What is the product of that reaction? We don't know? Amide, yes, thank you. Who said amide? You want a cookie? I don't have a cookie to give you. You like a piece of chocolate instead? Here we go. So when your tuition goes up because we have to buy more chalk, that's the man to talk to. Now you get an amide. You get nucleophilic carbonyl substitution. So enamines only happen Enamines only happen when there's no leaving group on the carbonyl, when there's only aldehydes or ketones. All right, so anyways, so the way this happens is actually a reaction that you're very close, well, hopefully you know. The only difference is one mechanism step in a reaction we've already talked about. What reaction have we talked about where a carbonyl compound, as I recall, was an aldehyde that we did in class, reacted with an amine to make a hint? Go back and look at your notes. Retinal, right? Amine plus, it was the lysine amine in opsin reacted with the carbonyl in retinal to make rhodopsin, make the imine. So that made an imine, carbon-nitrogen double bond. But the difference there, that was a primary amine, RNH2. This is a secondary amine. When you get to that very last step in the mechanism, and I encourage you to work out the mechanism for this process and see it, but when you work out the mechanism, you'll see it's a matter of whether or not there's a hydrogen on the nitrogen as to whether you're going to get an imine or an enamine. So there's our enamine that happens in this case. Again, work out the mechanism. And now at this point, you might be going, wait a minute. He's telling us to work out all these mechanisms because he's just rushing through this stuff at the end of the course and doesn't have the time to do it. Well, that's not true. Because I could say, the heck with the pharmaceutical chemistry, we're done today, but we're not going to do that, all right? The reason I'm doing it is because at this point in the course, you are beginning to climb or have already climbed or are near the top or maybe even falling down away from your third chemistry 14D transition state, your knowledge transition state. Your third one is predicting mechanisms for reactions that you've pretty much never seen before. You should be able to do that. So here are some really good practice examples for you, the kind of things we're working on there. And on top of that, these mechanisms are in the LCATSA chapters anyway, so you can just look them up if you, once you've struggled with them. All right. Now, what do we do with enamines? Enamines, like enols and enolates, are nucleophilic at the beta carbon. And actually, that's one of the primary reasons that they were developed for laboratory use, well after Mother Nature has used them for other things. But in the laboratory, there was need to do enolate kind of reactions, but in circumstances which for various reasons enolates couldn't be used. So this process was developed. So we can take an enamine, here's our ene, there's our amine, and react it with an electrophile, in this case an acid chloride. It is a carbonyl with a chlorine attached to it. So what does that make you think about in terms of carbonyl chemistry? Addition or substitution? Substitution. Chlorine's a leaving group. So there's your nucleophile. That can do substitution on the carbonyl. There's your carbonyl that came in. This guy is that group. But of course, the nitrogen goes away. That's what the aqueous H2SO4 step does. Once again, good mechanism for you to practice and work out. The mechanism is in LCATSA if you need it. But practice working it out first. Now, we've talked about three different related functional groups: enolate, enol, enamine. We might ask about their relative reactivity. They're all pretty similar in terms of the way they react. In all cases, we talked about nucleophilicity at the beta carbon. In other words, the carbon other than the carbon that has the carbon or the oxygen or the nitrogen. So here are possibilities: enolate, enamine, enol, and alkene. Now, they're in this particular order because of the following. Enolate has the formal negative charge. The enol and enamine are neutral. So enolate, formal negative charge, we know that more charge means more electron density, more nucleophilicity. Now, the other two structures that are listed, enamine and enol, the enamine is more nucleophilic than the enol because of electronegativity and resonance. In both cases, it is a pair of electrons that are being moved around through resonance. So here we have our atom X with a lone pair, whether it's oxygen or nitrogen. So we end up with a structure, a less important resonance contributor, but certainly a resonance contributor. This is one that explains why we have nucleophilicity out here on the beta carbon. But this is a resonance donation issue. So atom X, whatever it is sharing its lone pairs. The better X is at sharing its lone pairs, the more electron density gets pushed out here towards the beta carbon. So which atom is between an enamine, where this is a nitrogen, and an enol, where this is an oxygen, which of those two atoms, oxygen or nitrogen, is more inclined to share electrons? Nitrogen, because nitrogen is less electronegative, right? Less electronegative, more philanthropic with its electrons, more inclined to share. So an enamine is more nucleophilic than an enol. And I put alkene up here simply because it's really like those two other three things. It's got a carbon-carbon double bond in it, but it's a much weaker nucleophile because it doesn't have the lone pairs on the X atom to contribute the electron density. But other than that, these guys have some pretty similar reactions. Now, another question that gets asked at this point, before we talk a little bit about these functional groups in biology, is how do I know that, first of all, how do I know that a reaction may involve an enolate, enol, or enamine? And how do I tell, if it is one of those, how do I tell which is operating? Well, here's one thing to look at. Here's the clue we saw several times. If the reaction, whatever it is, occurs at the alpha carbon, and usually it's a case where we're taking a hydrogen away and putting something else on there. So if that reaction occurs at an alpha carbon relative to a carbonyl, that probably means, it's not a guarantee, but it's a pretty good indication that your reaction is enolate, enol, or enamine. Now I say pretty good because we know of at least one other class of reactions in which a CH bond can be converted into something else like that. What other class of reactions will do that? Well, close. Chi-chi-bobbin does replace hydrogen. It's a good guess, but that only happens at pyridine hydrogens. Good guess, though. What did we talk about after electrophilic aromatic substitution? Radicals. Remember? Free radical halogenation is a way to convert a CH bond through a radical process into a carbon-chlorine or carbon-bromine or even a carbon-fluorine bond. So you could do that next to a carbonyl. It turns out to be kind of a messy thing, but potentially I suppose you could do it. So if you've ruled that out and you know that it's either enolate, enol, or enamine, how do you tell between them? Well, enolates pretty much require strong bases to make them. So if there is a strong base present, you're probably dealing with an enolate mechanism. Enols require usually some acid to form the enol from the keto form. So if there is strong acid present, that is probably enol. Enamines require nitrogen. Enolates and enols do not. So if there is an amine present somewhere, a nitrogen atom present somewhere within your molecules, then maybe you're dealing with an enamine possibility. So those are a pattern to think about. All right, we're going to close up this topic today by talking about these molecules, these functional groups in biology, which is part of the reason we're talking about. I mean, obviously we spent a lot of time in this course talking about biological kind of applications. Now, enolates in their naked form, in their simple form, like acetone enolate, are generally too basic, too strongly basic to occur under physiological conditions. Why would that be? Yeah, probably 71% now that I've had my half a bottle of water here. But as she points out, we have a lot of water in us. Water is more acidic than a lot of carbonyl compounds. So the water will easily protonate simple enolates, like acetone enolate. Now, if it's a more stable enolate, if it's, say, an enolate between two carbonyls, water usually isn't an issue. But for a lot of the simple enolates, water is just too acidic. As soon as the enolate will be made, it would get protonated. However, enols and enamines are plentiful in biological molecules, either simply as part of the structure that goes on to do something else, or in some cases, because they're used for carbon-carbon bond formation. Here are some examples you're familiar with. Ascorbic acid, good old vitamin C, has an I feel like I'm being criticized from next door. Has an alkene ol, or it has an alkene ol, so you can actually view either one of those as being the enol. And tryptophan, we can think of as having, there's the amine next to the carbon-carbon double bonds. Now here you can think of it as being next to the benzene ring and count those carbon-carbon double bonds or part of the 5-membered pyrrole ring. And of course these are both, or taken together, these are aromatic, so whether or not you want to think about that as an enamine. But it does actually have some enamine kind of character to it, because the way this guy is synthesized involves making a new bond right here at this carbon, that puts this piece in, involves making a new bond here at the place where the enamine would be nucleophilic. And this tryptophan, by the way, this is the stuff that they tell you puts you to sleep at Thanksgiving, right? If you have too much, you eat turkey, it has tryptophan in it, it puts you to sleep. If that was true, almost everything you eat would put you to sleep, because tryptophan is a common amino acid. It's present all over the place. The reason that you go to sleep at Thanksgiving is because you ate too much turkey, drank too much beer, watched too much football, and were around too many boring relatives. That's probably the reason you fell asleep at Thanksgiving. All right, so what kind of things can we do here? Let's see, some biological processes. Well, one that you are involved in right now is making carbon-carbon bonds through a Claisen kind of reaction, except it involves thioesters. So remember, in a Claisen reaction, we take an ester, make the enolate next door, add that to another molecule of the ester. Here we're going to do it with a molecule called acetyl-CoA. It's a thioester. And other than that, it behaves pretty much like an ester. Frequently, this thing is used as a 2-carbon building block because the S-CoA, that's the leaving group. It essentially leaves as an RS minus. So there's an enzyme that causes this process to occur. And you can draw it out if you'd like as a Claisen condensation. It pretty much works that way. Make the enolate, add to another molecule of this. Now, and perhaps a more interesting process that's going on. Oh, this, by the way, is involved, and you stitch a whole bunch of these together to make fatty acids. And other things like squalene and so forth. So another example is glycolysis, which is the metabolism of glucose. And so here's glucose to begin with, and the end product of glucose, essentially, metabolism is essentially carbon dioxide. And to do that, you're going to have to take the glucose and break carbon-carbon bonds to make these smaller molecules. So how does that happen? Well, it starts with cyclic glucose, and through several steps, the thing is opened up and some phosphates are put on, but it still has all its carbon-carbon bonds. Now, to break it apart, the carbonyl here on the end is reacted with a primary amine on an enzyme because it is a primary amine. That's going to make the imine here. This is a protonated imine. And then another enzyme is going to take this apart. Now, how it goes from here to here, another good mechanism for you to work out, but it is a reverse aldol. Now, what do we mean by reverse aldol? In the forward direction, you make a carbon-carbon bond. The tetrahedral intermediate gets protonated, we move forward. Imagine that tetrahedral intermediate with a negative charge and the aldol reaction instead went backwards. If its leaving group that it ejected was the enolate, the net effect would be breaking carbon-carbon bond. And that's exactly what happens here. So I'd like for you to draw the curved arrows and try to see if you can figure out how this guy gets chopped apart to become that aldehyde through the taking apart process. Now, this molecule is useful. This is the other product, by the way, of this retroaldol. So we get two things, an aldehyde and an enamine. This guy is useful because it can be fed directly into the pyruvic acid cycle. It gets metabolized and so forth. This guy, on the other hand, there is no direct pathway to use it. I can't feed that into the pyruvic acid cycle. Now, that's a pity because that's 3 carbons, which is exactly what the pyruvic acid metabolism pathway needs. So it's not very green to throw away, not very efficient, if you will, to throw away half of glucose. After all, let's suppose you're a predator or even a plant eater. You've spent energy to get that glucose. If you're a cheetah, you chase down a zebra. If you're a brontosaurus, you had to reach all the way up to the top of the tree, whatever it is. If you're a student, you had to go all the way down to Ackerman to stand in line to get your Rubios, to get your glucose, right? But you expended energy to do that. Now, from organisms, it's all about conserving energy. You want to do the least amount of energy. That's why cheetahs, for example, don't chase everything. They look at the adult zebra, and especially the one that's got the gun, and say, no thank you, it's not worth my time. That's why they chase the sick ones and the baby ones, because it's less effort. They do. If you're a baby zebra, you're in trouble. It's Mother Nature. Mother Nature is mean and cruel and vicious. right? Didn't you know that? Yes. She's lazy too, but... So it would be nice, because this is 3 carbons, it would be nice to recycle that. On top of that, it's kind of close to this, because it's got, look at this, it's got a carbon with an oxygen, a carbon with a nitrogen, and a carbon with an oxygen. This thing is a carbon with an oxygen, a carbon with an oxygen, and a carbon with an oxygen. So if I could convert this into this, then I could use the other half of the glucose. Well, it turns out you can, because if you take this enamine and you hydrolyze it, it takes you to this molecule which is called hydroxyacetone phosphate. So there's the acetone. It's got 2 OHs, one of which is a phosphate. You can do that hydrolysis. An amine hydrolysis is something you ought to be able to do. And then another enzyme converts it to the enol, which can then be converted to the this guy, which is exactly what we wanted. So this thing can be recycled, so to speak, or at least converted into something useful through an enamine, keto, enol, back to keto kind of pathway. Good practice for you to draw out some curved arrows to do that. So Mother Nature is making use of those. All right. So at that point, we're going to be done talking about, finally, carbonyl chemistry. Now, the last thing we're going to talk about in this course is a little bit about, we're going to do drugs here. We're going to talk about a little bit about pharmaceuticals, and we don't have a whole lot of time to do this, so I don't want to really rush through it. So we'll cover as much of this as we can, and the final exam coverage of this material will take into account the fact that we're doing it fairly late in the course and the fact that there's not a whole lot for you to read on this particular topic. To give this topic a complete coverage, we would need several quarters. As you're probably aware, pharmaceuticals and how they interact with biological structures, there's an awful lot of biology and chemistry that's out there. Our main focus in this is just kind of to be an introduction to talk just a little bit about how this thing works, but we want to do it by emphasizing the fact that biological structures are really just chemical structures. So when we talk about how pharmaceuticals work, we're really just talking about how organic chemicals, the pharmaceuticals, interact with other organic chemical structures, proteins, receptors, enzymes, whatever they are. All right. So let's start by talking a little bit about what we're going to be talking, some vocabulary words here. Drug, of course, a drug is a substance which is introduced to do all sorts of things. Maybe you take it to alter, supplement, or block some chemical process in an organism. Now, maybe in you, or it may be inside an organism that is in you. Maybe you want to kill bacteria that are inside of you. When we talk about drugs, we're also talking about substances of abuse. We may also be talking about pharmaceuticals that are of clinical use, or pharmaceuticals that have both. Or we may be talking about other drugs that we don't think of as drugs. For example, caffeine. For how many of you, caffeine is your drug of choice? Right? It's a drug. You take it to stimulate your biological process. See, some people drink it just because they like the taste of it, but caffeine is a drug. Well, you don't like the taste of caffeine. If you've ever tasted pure caffeine, very bitter. You wouldn't want to eat pure caffeine. So your drug of choice might be coffee. It just happens to have caffeine in it. Now, the other thing we're going to talk a little bit about is pharmacology, which is, in broadly defined, the science of the study of the effects of these chemical compounds on living animals. Now, usually that means we're focusing on medicinal drugs. But also we're talking about the manufacture, we're talking about how they work. You can even arguably include sociological effects in this. Normally we don't, but you can include that as well. So when you go to pharmacy school, there's a whole lot of things you might study there. Now, so why do we take drugs for the most part outside of caffeine because it makes us feel good, or other drugs because they don't make us feel good? They used to at one point the first time we used them, but then they became, we're addicted to them, those kind of drugs, right? We're talking about medicinal drugs here. Although caffeine, captopril, if you're taking, probably nobody in here is taking captopril, I hope not. High blood pressure medication. It's one of the most prescribed, used to be one of the most prescribed drugs out there. Whatever drugs you are taking, right? We're going to focus mostly on drugs that deal with, that we would think of as pharmaceuticals, drugs that deal, not freelance pharmaceuticals. Right? That's the guy in the corner that goes, "Psst, need to score a nickel bag?" Those are freelance pharmaceuticals. Freelance pharmaceuticals is what keeps the TV show Cops in business, right? Right? You know what I'm talking about, right? You been on Cops? No? So I don't have to, because when we watch Cops at home, right, as soon as, little sidebar here, we love Cops in my house, right? We have a Cops drinking game and everything, right? Actually, it's an M&M game. We don't, because if there's many things as you would drink for on Cops, you'd be, the first vignette and you'd be done. But my wife will tell you, as soon as somebody starts resisting arrest, I go, tase him, tase him. All right, anyways. So we're going to talk about disease states. We have to think a little bit about what are disease states, what do we seek to do with the drug? In other words, we need to understand what the chemical basis of the disease is so we can figure out what kind of pharmaceutical we might take to deal with that disease. Now, there are many, 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 many different kinds of diseases, as you're probably well aware, but here are just a few. All right, first of all, we might have a deficiency of some sort of critical biochemical substance. In other words, whatever it is that we need, the body doesn't make enough of it. Solution? Usually, it's to supplement it, what we call replacement therapy. Now, you can sometimes stimulate the production of that biochemical. That's one way to do it. But another way to do it is supplement. Now, replacement therapy, diabetes is probably one of the biggest issues that usually comes from a lack of insulin. So you can take insulin, and that helps resolve that issue. Hormone replacement therapy for menopausal women has been a big deal lately in the news as well. You might have the other aspect of the other end of the spectrum, and that is you might have too much of something. Well, it's usually not easy to take it away, and so instead, because it may be spread out over your entire body. So instead, the idea is you simply suppress its reduction, you slow it down or stop it, and eventually the body will reach equilibrium with that. So a very common example, especially this time of the quarter for students, is the production of excess gastric acid. In other words, tummy trouble. So what do you do with that? One of the things you can do, there's a whole bunch of solutions now, but the most common one is to suppress production of stomach acid. And there are a slew of drugs that do this that have started showing up in the last 3 years. The biggest one, the one that really started it all, was this guy, cimetidine, also known as Tagamet. When Tagamet came out in 77, it was the first drug to sell $1 billion, that's with a B, dollars. First year out, $1 billion. And then when it went off patent, eventually, SmithKline lost their market share, and they still make a whole lot of money off it. But this is stuff you can buy now. I don't even think it's over the counter anymore. I think you should just walk up to the shelves and get it. But that was a big deal because it was the first drug that did that. Now there's literally probably 100 different drugs that do that. Something else you may have to deal with is invasion of an alien organism, i.e. an infection of some sort, and this alien organism produces some sort of toxic substance. The solution? Kill the organism. Kill the bad organism, right? Don't kill the host, kill the bad organism. And a good example of that is botulism, which comes from this little bacterium. And so what it does is it produces a protein toxin, and it's kind of hard to get rid of the protein toxin because if you get rid of some of the toxin, the bacteria kind of shrugs its shoulders and makes more. So the idea is to kill the source. You kill the bacterium. Then the question becomes, how do you kill the bacterium without killing the host? Well, target things that only bacteria have. Then, of course, one of the most frightening forms of disease these days is uncontrolled cell growth, i.e. cancer. And the solution, now there's lots of ways have been developed to potentially target cancer. But one is to target processes that are essential to, or perhaps just faster in reproducing cells. For example, DNA reproduction, because cancer cells are growing much faster than the other tissue around them. So if you target things that the cancer cells are doing faster and kill it when that's happening, if you say, aha, you're replicating, so now I'm going to have a drug that's going to target you and kill you, chances are you're going to kill cancer cells faster than you kill normal cells. But the problem with this strategy is cancer cells have a lot of processes that regular cells do. And so if you target those cancer cell processes, eventually there's some toxicity in regular cells. That's part of where chemotherapy toxicity comes from. There's a number of things you can do to interfere with cancer, interfere with DNA replication we talked about, or a relatively new strategy is to interfere with angiogenesis, which is the growth of blood vessels. And as it turns out, because tumors are growing, because cancers are growing more quickly than the cells around them, the cancers have a greater demand for blood growth, as a matter of fact, or blood vessel growth. Some of them even produce signals, hormones that go out into the surrounding tissue and say, hey, we're growing, build blood vessels. And if you can block those processes, if you can prevent the blood vessel growth, then maybe the body will have an opportunity to take care of the cancer on its own. This is actually how thalidomide works as an anti-cancer. Remember we talked about, if you remember back in 14C, we talked about thalidomide, and it was a late 1950s, early 60s drug that was given to hysterical, as they said in those days, pregnant women, right? They were hysterical, running down the street screaming. Actually, no, they were just moody. I don't even think we use that word anymore either. I don't forget what the correct word is that we use. What's the politically correct way to say they're moody? I know there's an unpolitically correct way to say it, but anyway, so that's what thalidomide was given for, but thalidomide caused an awful lot of birth defects, shortened arms and or shortened legs. So it was taken off the market, but after a while they figured out what was going on. It was interfering with blood vessel growth, and somebody said, hey, wait a minute. That's a potential way to treat cancer. So thalidomide is actually now back on the market as an anti-cancer drug. It's not the number one anti-cancer drug, but it is one therapy. Okay, so those are some of the kinds of things we might want to target with drugs. Now, how do we do it? We need to talk a little bit about the chemical basis for how drugs work. We're going to be very simplistic about this because, after all, we're dealing with incredibly complicated chemical factories. A cell has something like 50,000 different chemical processes going on at any time, and you have to figure out how to target the one or the few that you want to target without messing with other things. So we got to ask, what's the chemical basis for drug action? What kinds of biological structures do we need to think about in terms of a chemical effect? And what's the relationship between how those structures work and the activity? Why are some molecules really effective pharmaceuticals, but if I add a methyl group, they completely shut down in terms of drug effect? All right. We're going to start by talking about the two broadest classes, and that is receptors and enzymes. So receptors are proteins that basically kind of are the doorbell for some other process to start. So in other words, when a receptor, which is the protein, when it binds some other molecule, and that other molecule is called the signal molecule, this binding process sends a signal which tells some other process to happen. For example, olfactory receptors in your nose. When you smell something, when that molecule, whatever it is, docks with the olfactory receptor, if it's a good dock, if it's a good match, the nerve attached to that goes, aha, and fires, and a signal goes to your brain, and your brain goes, oh, I've smelled whatever it is you've smelled. So that's a receptor. That's how that process works. Now, another example is a neurotransmitter receptor, and this basically is a receptor on a neuron that when the signal molecule comes in, it tells the neuron, okay, it's your turn to send the signal downstream. If you block that receptor, if you make it physically impossible for the signal molecule to come in, then the receptor isn't triggered, the signal isn't sent. In this case, it means the neuron doesn't fire, and so you've blocked whatever the nerve's response is. Maybe it's a pain nerve, for example. Now, in a cartoon fashion, this is kind of how this happens. Here's our receptor, and there's the signal molecule, and they're drawn in a way in which the shapes are complementary. That is important, because you don't want your receptor to dock with any old molecule and signal. Imagine, for example, the receptor you're talking about is the olfactory receptor for pick a smell. Cinnamon, thank you. Were you thinking cinnamon?
Sure.
Sure, what the heck, right? Cinnamon. Imagine anything triggered the cinnamon receptor. Cinnamon, 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 right? Your brain would go nuts. Imagine, for example, it was a pain receptor, right? Oh my God, aldol reaction, pain, Claisen condensation, pain, everything caused pain. You need some selectivity. So the selectivity comes from the fact that the receptor's cleft, active site, whatever you want to call it, has a specific size, a specific shape, and specific amino acid chains, for the most part, hanging in there that the signal molecule has to agree with. The shape has to be right. So for example, if the receptor is looking for a long, skinny molecule, then a long, skinny molecule has to dock. A short, fat molecule probably won't fit. If the receptor is loaded with lots of OH groups, so it's a very polar receptor, it likes to bond through hydrogen bonding, then a signal molecule which doesn't have hydrogen bond acceptors and or donors isn't going to be accepted, and it won't trigger it. So that selectivity is really important. It's one of the most fundamental ideas if you're designing a drug to hit a particular receptor or enzyme. You've got to make sure your drug is going to be docked. All right, so once this thing docks, then we get the signal receptor complex, and that process, the process of docking, is when the receptor sends off and it says, yes, you're the right signal molecule. The receptor sends off its signal and says, aha, whatever process it is, I'm going to signal for that to happen. Now, you don't want the receptor to be constantly bound to the signal molecule, because either the receptor might continue to fire, which may not be a good idea, or the receptor may only fire once to be done with. So imagine your olfactory receptor bound whatever molecule is. The cinnamon olfactory receptor bound that molecule. Once when you were 2, you went, cinnamon. And then for the rest of your life, you couldn't smell cinnamon anymore because it was that molecule, whatever it was stuck in the olfactory receptor. It needs to leave. It may do it quickly, it may do it slowly, but it needs to leave to free the receptor to do its business again. Now, when we bind to a receptor, 2 different things can happen. You can either trigger the receptor or not. And depending upon what your drug is trying to do, you've got to tune it to do that. There may be a circumstance where there's a lack of a biochemical that you need, and so you add more of this stuff and you want the receptor to trigger. So maybe you don't have enough neurotransmitters in your brain, then whatever drug you're putting in, you want to cause that neurotransmitter to fire. Or let us suppose it is pain receptors that are the issue, and maybe you're very sensitive to pain because your pain receptors are constantly accepting signal molecules and constantly firing. You'd like to block those receptors by putting a molecule in there that keeps the natural signal molecule out, but you don't want the signal or the receptor to fire. So there are 2 classes of groups of drugs that we talk about. There are antagonists that have the strong affinity for the receptor. So they bind well, but they don't actually cause the receptor to fire. Then there are agonists, which also have strong affinity, actually often stronger than the natural substrate because we're trying to get the receptor to fire, and these things cause the receptor to fire when it's bound. Here's a couple of examples. Give me just 30 seconds and we'll finish up this page. Curare is the poison dart stuff. So if you ever go down to Brazil and you get lost in the jungle, and the natives say, hey, there's a person that looks like we'd like to eat them, so they blow their little darts at you and then, make a nice stew out of you, a little parsley, a little cilantro. Right? When they blow the poison darts at you, the molecules in the curare, the one in particular, whose name I forget off the top of my head, causes paralysis by antagonizing, binding with but not firing neurotransmitter receptors. Basically, your breathing stops. Then there is Pepsid, also called famotidine. This is a gastric acid suppressant. It targets histamine receptors. Now, histamine, the same histamine that makes you allergic, also triggers the gastric acid secretion in your stomach. And if there's too much histidine, if there's too many of the receptors firing, you get lots of gastric acid, you get an upset stomach. So this stuff works by suppressing gastric acid suppression. It targets the histamine receptors. It blocks them, another antagonist. Prozac, on the other hand, also an antagonist. It's a serotonin uptake inhibitor, and it blocks the receptors that normally would re-accept serotonin, which would cause all sorts of issues in your head. All right, next time we'll start talking about, on page 70, we'll talk a little bit about enzymes. See you on Wednesday.